Hello, I'm Professor Fan, and in this video, I will be performing a spatial structural alignment between a single-cell resolution Xenium spatial transcriptomics dataset and a spot resolution Visium spatial transcriptomics dataset from serial sections of a breast cancer tumor using our lab's tool STalign in order to facilitate downstream spatial molecular comparisons between these two different spatial transcriptomics technologies. So for this tutorial, we'll be using some publicly available spatial transcriptomics datasets from 10x, though this tutorial will be applicable to any spatial transcriptomics data for which we have single-cell resolution information, either by way of cellular positions or registered histology image. I'm just downloading the necessary files, in this case for a spot resolution Visium dataset. I'm tarring and organizing my files in a nice folder and double checking that I have the spatial information I need and also the gene expression information. And of course, likewise, we can do that for our second Xenium dataset, just double checking this is the one I want. I think there's a couple datasets available. Okay, this one looks good. And again, just downloading the necessary files, which in this case is pretty large file. Let's just untar that. And again, organizing our files for later and double checking that I have the gene expression information and also the cellular position, which maybe, it's probably in the cells.csv file. Okay. Again, we'll use STalign, which is a Python tool my lab has developed for pairwise spatial alignment of such spatial transcriptomics datasets using diffeomorphic metric mapping. STalign is implemented in Python since it uses PyTorch for some stochastic gradient descent. I've already installed STalign via pip, so I'm just going to import it along with all the dependencies. And now we will read in the single-cell resolution Xenium dataset first and update the file paths. I'll be copying pretty heavily from this tutorial. And we can plot the positions, again, updating the column names appropriately. And this looks good. Each point is a single cell, and we have their positions in the tissue. And now we can create a rasterized image. STalign focuses on image alignment, so we have this image-based representation of our spatial transcriptomics data. And we'll refer to this as the source image, and I'll just normalize that image to make sure the intensities go from 0 to 1. Okay, and now we can do the same thing for the Visium image via its H&E stain. Again, I will normalize the image intensities, double check my image dimensions, which are a little bit different here. And I will refer to this as our target image. And of course, we can visualize these source and target images side by side. In this case, these are two data sets that come from adjacent serial sections of the same breast cancer tumor. So they're not profiling exactly the same cells, but of course, the overall structural, our ductal structures look quite similar, so we can perform a spatial structural alignment. But as you can see, they're not oriented in the same direction, and even if we try to manually rotate one of the tissue sections or one of the data sets, you can see how the coordinate ranges are a little bit different, and it's not always clear how much to stretch or rotate. So let's export these images so we can use STalign's point annotator tool to manually place a few spatially matched points to guide our alignment. Now I'll just download the point annotator tool, and it's a Python command line tool. So I'll just fire up a terminal and follow the tutorial instructions to place 3 spatially matched landmark points In this case, I'll give them names of duct 1, enter, duct 2, click, and place my points. Boop. Boop. Enter. Duct 3, place my points. Boop. Boop. Enter. Okay, that looks good. And it saves these as MPY files. So now I can read these points back in, and if I get the file names correct, whoops. Then we can visualize these points on our source and target images. And that looks pretty good. It doesn't have to be perfect, just enough to help orient and initialize a rotation and scaling via an affine transformation. So now we can actually use STalign to solve for a set of rigid rotations, translations, and other affine transformations by minimizing the distance between the landmark points that we just placed. And of course, we can apply this learned transformation to our source image to double check that it did indeed perform the expected rotation and scaling to orient the tissue in the same direction as the target. And of course, we can apply this transformation to our single cell positions from the Xenium to move them into a shared coordinate space and visualize these cells overlaid onto the H&E image from the Visium. And I think visually this already looks pretty good. Again, these are serial sections, so the cells are different and they'll never match perfectly, but there still seems to be some very minor structural differences due to maybe local distortions in the tissue, or maybe I just didn't place my landmarks perfectly. So let's try running a diffeomorphic component of the alignment now. The diffeomorphic alignment component has a number of tunable parameters. What STalign is trying to do is minimize the distance between the source and the target image by minimizing a particular objective function that has many parameters. So we can read the documentation or the online supplementary notes of the original paper to learn more. Always good to read the documentation. But for now, I think I'll just try using the same parameters as the tutorial, but decrease the number of iterations just to try things out. There are many tutorials available from different tissues, so it's good to check them out. Many of these parameters do have the same units as your XY coordinates, so if a tissue is really big or really small, these parameters will need to be tuned accordingly. Okay, in this case, it looks like I was able to run without any errors. The smoothing kernel looks reasonable. The contrast transform source looks more like the target. So I think we're all set for running this with more iterations. I am running this on a laptop CPU, so it will take a few minutes. It'll definitely be faster with a GPU, but I guess we can always take this opportunity to grab a tea, or better yet, to pet the Remy. Oh. Now that our alignment is complete, we can just go back to the tutorial and pull out the learned velocity fields and other transformations. And then we can apply our learned transformation to the original single cell positions to move these single cell points into the aligned coordinate space. And of course, we can plot these new aligned single cell positions onto our Visium H&E image. Okay, so I think the alignment can still be improved, particularly towards the top right corner here. I often do tinker a lot with parameters, and we'll try aligning many times. So I'm going to give this one more shot and speed things through.
20 minutes later.
Okay, so now we see a decrease in our error, and let's try to look at this again. And yeah, I guess maybe this looks well aligned enough to me. Let's write out the aligned spatial point coordinates and save them for downstream use. And yeah, I've appended the aligned positions, and I'm going to save it as a csv.gz file. Okay, so to double check everything, why don't we read in the Visium spot positions and overlay them with the H&E image, which should already be registered. But of course, now we have the new aligned Xenium single cell positions too, so we can plot everything all together. And I'm just going to first try to plot the Visium spots on the Visium H&E image. But in this case, the positions are not necessarily in the same scale. We have to take into consideration the scaling factor because of the resized histology H&E image, so be sure to incorporate that. Okay, this still looks a little bit funky. I think I always swap my rows and columns. It's X and Y, but rows and columns. Let's just double check with the online site assist. And in this case, this looks, I think this time I got it right. The spots are indeed mapping or plotting onto the expected region in the tissue. So for the sake of demonstration, I'm just going to try to subset both the Xenium and the Visium datasets into a shared region of the tissue based on their aligned XY coordinates. Maybe I'll first just try plotting. So this is a small region of the tissue for which we have both Visium spots and Xenium single cells. And maybe I want to make the tissue, the shared region, a little bit bigger. Oops, too big. Now we've fallen off the Visium. I think this is pretty good. And why don't we now subset to this particular region for both the Visium and the Xenium datasets so that we can use the subsetted aligned data for some downstream molecular comparisons. Okay, I'm just going to skip through some of my struggling with Python and try to get to the end results. And it's always good to visualize to make sure we are indeed now subsetting to our shared region of interest for both the Visium and the Xenium datasets. And now we can write out our results and save them for downstream use. Mm-hmm. Okay, so I am actually personally much more comfortable and fluent in R, so I will actually switch over to R and read these aligned and subsetted cells and spots into R to focus on downstream analysis and visualizations there. Always good to take a look at the subset of your data and also to visualize it. And in this case, well, we can actually also visualize things together. Of course, the single cell density is quite variable, and we can see these nice structural variation while the spots are kind of uniformly gridded. But why don't we read in then the gene expression information next? The gene expression information is stored as these sparse matrices, so we can read them in using the matrix package. The row names and the column names are going to be stored separately. So I will also read these in as these TSV files. Oops, be sure to double check that you have your headers and delimiters appropriate. And of course, we can subset to the same set of cells for which we have the aligned positions based on their cell IDs. Now we can see that we have gene expression for a matrix with 541 targeted genes and 88,591 single cells. And let's just plot these cells using ggplot and color them by their total genes detected per cell. Maybe I want that in a log scale. Maybe I want to choose a different color scheme. Always lots of options with ggplot. Okay, and we can do the same for our Visium dataset. Again, reading in our sparse matrix and reading in the barcode names and row name or feature names, gene names. So note here we have 18,085 genes due to the untargeted full transcriptome profiling, but only 3,211 spots. And now we can plot these spots and color them by their total gene expression detected per spot. And we can begin seeing some similarities in terms of the underlying structural variation because genes are going to be more highly detected in some parts of the tissue than others, even though the spots are uniformly gridded everywhere. And we can even see how shared genes profiled by both of these technologies are spatially distributed, like epidermal growth factor receptor, EGFR. And we can definitely see some similarities. It'll be good to write some additional code to quantify these similarities and differences. Okay, so now we've aligned our single-cell Xenium spatial transcriptomics dataset to our spot resolution Visium spatial transcriptomics dataset of these two different serial sections of breast cancer tumor using STalign. And now we can begin quantitatively comparing, for example, the detection sensitivity of different genes at shared spatial locations across these two different tissues from these two different technologies. Or we can use physical alignment as a way of imputing gene expression or enhancing resolution to leverage the unique strengths of these two different spatial transcriptomics technologies. But all that will be left as an exercise to the students. Thanks for watching. If you've made it this far, I hope you got the chance to download the data and code along to try things out for yourself. For more details on STalign, check out the original published paper along with other tutorials and resources on our lab website.